Ting earned his PhD from the University of Michigan, and he subsequently conducted postdoctoral research at the UT Southwestern, and then he joined the National Institute of Biological Science in Beijing. His laboratory focused on two complementary research areas, the discovery and the development of the molecular glue degraders, and then identification and validation of novel therapeutic targets for the cancer treatments. And always the molecular glue seems to find Ting in different flavors, and we are very much looking forward to your seminar. As of note, today we'll have a question and answer session at the end of the second seminar. Thank you very much. Ting, the floor is yours.
Yeah, let me get the pointer. Okay, I guess the cursor, people can see it. So first of all, I'd like to thank Mikawai for the invitation to speak at this wonderful webinar series. Although YouTube is blocking China, but I always find a way to view these recorded online seminars. For those of you who are unfamiliar with me, I lead a research group at the National Institute of Biological Sciences, and also affiliated with Tsinghua University. And our institute is located in Changping, Beijing. So anyone in the audience are interested in visiting me and our institute, please contact me. And my lab is primarily focused on addressing the drugability challenge, as illustrated on this slide. Currently, only about 20% of our genome have drug-encoded proteins that contain druggable pockets that are suitable for small molecule binding. The remaining 80% of proteins lack well-defined ligand binding sites, and therefore often considered as undruggable, which is a great challenge to the reach of to realize the potential of conventional small molecule-based pharmacology. Over the past 2 decades, targeted protein degradation has emerged as a paradigm shifting strategy that overcomes the aforementioned limitation by inducing proximity between E3 ubiquitin ligases and target proteins, promoting the ubiquitination and subsequent proteasomal degradation of target proteins. Moreover, the rich diversity of E3 ubiquitin ligases have been believed to be a goldmine for TPD applications. Two main classes of small molecule degraders have been materialized. Molecular glues are monovalent compounds that stabilize pre-existing weak protein-protein interactions. In contrast, PROTACs are rationally designed bifunctional molecules that connect a E3 ubiquitin ligase ligand with a ligand to a target protein with a linker. Despite these two modalities have their own advantages, they also have their own disadvantages. Molecular glues are largely discovered by phenotypic screening and by serendipity, and usually in a target agnostic manner, therefore limiting their potential application in the industry drug discovery effort. In contrast, PROTACs approaches currently are limited by dominant reliance on two constitutively active E3 ubiquitin ligases, including cereblon and VHL. My entrance into the field of targeted protein degradation is also quite serendipitous. Over the past decade, we have been conducting phenotypic screening to identify bioactive molecules, followed by target identification and deconvolving their mode of action. I sort of like to highlight the advantages of this approach. First, chemicals can act quickly and reversibly, therefore potentially, potentially providing a strategy to uncover novel biology that are not revealed by conventional genetics. Second, target identification often can reveal druggable pockets that we did not know. And if these pockets are amenable, these pockets can be amenable to target-based drug discovery. And last but not least, the molecules we identified from the drug-like libraries often provide chemical starting point for further drug development. This simple strategy has surprisingly resulted in the serendipitous discoveries of 3 molecular glue degraders. Together with work for many other labs, these discoveries have sparked a lot of attention and interest in the field of molecular glue degraders. The first molecular glue I encountered is called inducilam. This molecule is a compound discovered by ASI in a phenotypic screen for small molecules with anti-cancer activity. During my postdoc training with Deepak Nichawan at UT Southwestern, we collectively discovered that inducilam acts as molecular glue to induce a ternary complex between RBM39 and a substrate receptor called DCAF15. And we further show that the level of DCAF15 expression can predict drug sensitivity. And this set of biochemical and genetic data were confirmed by structural studies from Eric Fisher and others, basically confirming the molecular glue mode of action. And more recently, these efforts collectively have enabled the re-entry of RBM39 degraders into clinical trials with appropriate patient cohort biomarker with clear biomarker-driven patient selection. After the inducement work, I started my independent research program at NIPS, Beijing. We conducted another high-throughput reporter-based screening and serendipitously discovered a molecule called HQ461. And this molecule induced the interaction between a kinase called CDK12 and a adapter protein of the Cal4 ring ligase, which is DDB1. And this ternary complex actually converts CDK12 into a substrate receptor of the Cal4 ring ligase, leading to the polyubiquination and degradation of CDK12's binding partner, cyclin K. At the same time, molecular glues with the same mode of action, but with diverse structures, were reported by the groups of Ben Eber, Nico Thoma, Jörg Winter. Further structural work from Nick Thomas' group reviewed the design principles of these diverse cyclin K degraders. CDK12 has a C-terminal extension that inserts into the DCAP binding pocket of DDD1. And molecular glues actually stabilize such pre-existing weak interaction, which have already been seen as the perfect complementarity between DDB1 and the CDK12. For the rest of my talk, I'm going to primarily focus on the third molecular glue discovered by my group. This work is an outcome of collaborative efforts from 3 graduate students from my lab and Pan Rui, Yalong, and Lei, and a graduate student from my colleague Niu Huang's lab, Xin Tongren. Again, we started with a phenotypic screening without the goal to identify molecular glues. Because the other half of my lab work on anti-tumor immunity, we realized that interferon gamma is a master cytokine with anti-tumor activity in the tumor microenvironment. We therefore initiated a screening campaign to identify molecules with enhanced cytotoxicity against cancer cells in the presence of interferon gamma. We designed a sort of clever imaging-based assay and screened approximately 80,000 compounds and identified 3 related molecules, including the antipsychotic drug acepromazine, which preferentially kills cancer cells when they are exposed to interferon gamma. We further observed that the enhancement of ACE cytotoxicity can be observed when cells are exposed to either interferon gamma or interferon beta, but not to tumor necrosis factor alpha. Moreover, this interferon enhanced cytotoxicity is observed in only a subset of cancer cell lines, as shown on the left column, not in some other cancer cell lines shown in the middle, and also not in a variety of normal cells we could get, such as keratinocytes, NK cells, and activated T cells from human donors. We first focus on the cell line selectivity of ACE promethazine. Cutting a long story short, we found that ACE-sensitive cancer cell lines actually expresses very high levels of AKR1C, 1, 2, or 3, which belong to the superfamily of aldoketo reductases. By mass spectrometry, we found that these sensitive cell lines can reduce the ketone into a alcohol and adding 2 Dalton to the molecular weight of ACE. While in insensitive cell lines such as HCT116, they express a lower level of this metabolic enzyme. Therefore, ACE is maintained as its parent molecule. For those who are good at chemistry, you will realize that this conversion creates a chiral center. We therefore separated the mixture into 2 pure enantiomer fractions, and found that only the S enantiomer actually is active, while the R is not so is inactive. Next, we try to identify the target of SASOH. We conducted a pooled genome-wide CRISPR screen, a knockout screen, to identify genes required for SASOH-mediated cytotoxicity. Among the top enriched hits were nearly all the genes of the interferon gamma pathway, including the receptor, the kinase, and the transcription factor. In addition, we identified a gene called TRIM21, which actually is the expression which is actually induced by either interferon gamma or interferon beta. This observation led us to hypothesize that maybe the observed interferon synergy comes from the induction of TRIM21 expression. And this is indeed the case, because if we knock out TRIM21, we see a loss of toxicity. And if we overexpress TRIM21, we actually see enhanced cytotoxicity, consistent with the finding, consistent with the idea that interferon induction of TRIM21 actually mediates the anti-cancer effect of ACE. So what is TRIM21? TRIM21 is a bifunctional protein. It's both of E3 ubiquitin ligase as well as a antibody receptor in the cytosol. Biochemically, it's composed of a ring domain at its N-terminus, followed by a B-box coil-coil domain to mediate dimerization, as well as a C-terminal pi-spray domain, which mediates the recognition of the Fc region of antibodies. And functionally, TRIM21 recognize and degrade antibody-bound viruses that have entered cell through endosome escape. And upon recognition of antibody-bound virus, TRIM21 is activated and adds polyubiquitin to antibodies and viruses, leading to the neutralization and degradation of the infectious virus. Because TRIM21 is a E3 ubiquitin ligase, we naturally decided to look at whether some new substrates get degraded upon exposure to the molecule to the compound. We used label-free quantitative proteomics to compare to identify several proteins, most notably NAP35, NAP155, SMPD4, and GLV1, to be depleted rapidly, like 8 hours after compound exposure. These proteins are localized to the nuclear pore complex. And by Western blotting, we confirm that the S enantiomer actually is active at degrading these identified proteins, consistent while R is inactive, consistent with the distinct cytotoxicity profiles of these two compounds. The nuclear pore complex is composed of about 1,000 building blocks assembled from copies of about 30 different proteins and organized as three stacked rings: the outer ring, the inner ring, and the nuclear ring, flanked by the cytoplasmic filaments facing the cytosolic side and the nuclear basket facing the nuclear side. By taking a time course detailed analysis of protein degradation of each nuclear pore components, we found that the inner ring proteins of the nuclear pore complex was depleted at the first responder, followed by the depletion of subunits from the outer ring. Because nuclear pore complexes are the essential gateways to govern nuclear cytoplasmic trafficking, we wondered whether the cytotoxicity of ACE comes from the degradation of nuclear pore. So to make this argument, we need to find a way to block the recognition or the degradation of nuclear pore proteins by TRIM21 and see whether those confer drug resistance. We therefore employed a recently described method called CRISPR suppressor screen invented by Brian Liao's lab at Harvard. And we sort of bravely expanded this method into simultaneously profiling drug resistance across 30 genes by designing every single sgRNA with available PAM to tile the 30 genes of the nuclear pore complex, resulting in a pooled library. These sgRNAs essentially enable the generation of diverse nuclear pore protein variants by the Cas9 nucleus. The transduced cells were then cultured in the presence of either Vico or ACE, and for 2 to 3 weeks to isolate cells which become more resistant. And magically, this screening worked. We identified a hotspot in the autoproteolytic domain or APD domain of NAP98, which seemed to have enriched sgRNA count in this population. Partly based on the genetic evidence and some other cell biology experiments, we proposed a model in which TRIM21 actually is recruited to the vicinity of nuclear pore by recognizing the autoproteolytic domain of NAP98. This actually brings TRIM21 to the vicinity of the inner ring, particularly its ring domain, triggering the polyubiquitination of the inner ring components. And the subsequent degradation of other nuclear pore proteins could be a secondary consequence of structural destabilization of the inner ring proteins. The cellular activities I have been describing so far suggest that ACE-OH could be a molecular glue. To test this hypothesis, we proceeded to reconstitute the compound-induced ternary complex in vitro. We used isosomal titration calorimetry and found that we could not detect an affinity between SSOH with the APT domain of NAP98. However, when we titrate ASOH into TRIM21, we observed a modest affinity at about 16 micromolar. How about the two proteins? TRIM21 and naptonate, when we do the ITC titration, we could not detect a high affinity. But through other more sensitive assays, which I don't have time to show, we actually see these two proteins indeed display some basal affinity to each other. However, if we titrate the APT domain of NAP98 into TRIM21 in complex with SSOH, we got a KD of 600 nanomolar. So this thermodynamic behavior is highly consistent with a molecular glue mode of action. Next, we to examine the structural details of how actually the compound interacts with TRIM21 and likely with NAP98. The C-terminal pre-spray domain contains a pocket that binds to the FC region of TRIM21. So by mutagenesis and also binding assays and in the end co-crystallization, we found that the molecular glue actually sits at the same pocket of the that recognize the FC region of antibodies. This co-crystal structure also revealed a possibility of converting molecular glue into heterobifunctional PROTAC-like molecules, which we named them as TRIM21-directed PROTAC or TrimTACs. We linked ACE with either JQ1, which is a high affinity ligand for the bromo domain of BRD4, and also linked it to SLF, which is a ligand for FKBP12, so that this can be used as two compounds to study model protein degradation. Then we use these TrimTACs to examine whether they can induce the degradation of reporter fusion proteins. So we found that proteins like CDK4 and the BTK, which are diffusibly localized, could not be degraded by TrimTACs. In contrast, proteins which are forced to localize into multivalent and phase-separated compartments, such as PML, Cajal bodies, or nucleolus, are sensitive to TrimTAC-based degradation. Moreover, we conducted side-by-side comparison of TrimTAC with more conventional cereblon-based PROTAC. And again, we see that a diffusible reporter are only degraded by the cereblon-based PROTAC, but not by TrimTAC, while a multivalent phase-separated target are sensitive to both degraders. These observations actually highlight the unique properties of TRIM21 activation, which have been beautifully figured out by work from Leo James and William McEwan's labs. TRIM21, although it's a dimer, but it's an inactive dimer. Upon recruitment to monomeric substrate, this inactive dimer is still inactive, so it's not able to ubiquinate the substrate. However, if the substrate are in a multivalent state, multiple TRIM21 will be recruited and creating a chance for its N-terminal ring domains to form dimers. And this dimer is actually critical for interacting with E2 ubiquitin and to catalyze the transfer of ubiquitin to neighboring target proteins. These studies collectively made a convincing case that TRIM21-based degraders, either molecular glues or PROTACs, can be tools to selectively degrade proteins in multimeric assemblies. Such assemblies include like phase-separated condensates, such as oncogenic condensates, or higher-order assemblies in the immune signaling, as well as amyloid-like aggregates in neurodegenerative diseases, as well as viruses that have been replicating or have invaded the cell. As a quick proof of concept, we tested whether TrimTAC can be used to target C-gas And cGAS actually is a sensor of cytosolic DNA and when bound to DNA form condensates. Upon DNA transfection, we see cGAS forms a nice puncta, and this puncta can be degraded by TrimTAC treatment, while the unstimulated cells, cGAS is diffusible and not sensitive to TrimTAC. And in the end, we wonder like other molecules, because we know TRIM21 expression can influence TRIM21 dependent molecular glue activity. We wondered whether there are other TRIM21 dependent molecular glues out there with available phenotypic data. So by analyzing the PRISM data from the Broad Institute, we actually identified two compounds with more potent molecular glue activity. And notably, Drew's group used a similar approach and made identical discoveries. So what truly amazed us is that these structural diverse molecular glues, including another one described by Hidjan recently, they all promote the interaction between TRIM21 and NAP98. So they work exactly the same mechanism. So the precise design principles and the structural basis will become clear once we obtain high-resolution co-crystal structures. In the future, we would like to explore the diversity of the TRIM family, which comprises over 70 members in humans. They all have these very typical structures with diverse C-terminal domains, mediated substrate recognition. They have diverse functions, rich biology in the literature, but so far have not been heavily exploited for TPD. So which I think is a great opportunity for future investigations. I'd like to conclude by thanking my lab members and collaborators for their contribution to the work I presented today. I also acknowledge the core facilities at NIPS for their support and the funding agencies for their generous support that make this work possible.